I'm so excited to be able to talk about the book I have just written, and which is called Good Genes Gone Bad, published by Penguin House. We're going to talk a little bit about the book and see what we can reveal or unveil on the topics that I've written about. So for that, I'd like to invite my sister, Dr. Anjali Rao, who's in Bangalore, and Dr. Lieutenant General Madhuri Kanitkar, who's a dear friend from high school. The three of us are going to have some conversation. So I'm going to hand over to Anjali to moderate the session and the discussion within the three of us. So over to you, Anjali.
Thank you very much, Naren. A good day to everyone and a warm welcome. We are going to have the launch of the book Good Genes Gone Bad, written by Dr. Narendra Chirmule and published by Penguin. I'm Dr. Anjali Rao, and I retired from the central government health scheme as a physician and administrative head of CGHS Bangalore. I look forward to having a lively conversation with my good friend, Dr. Madhuri Kanitkar, who is a serving Lieutenant General in the Army. She and I share the same alma mater, which is the Armed Forces Medical College, Pune. Good to talk to you, Madhuri. Please give your details.
Thanks a lot, Anjali. It's such a pleasure. And thank you, Naren, for inviting me onto this wonderful panel of Good Genes Gone Bad. I think it's an amazing opportunity of getting to talk a little bit about the book, but more so important, an interaction and thought process on what went behind this book, because I think that's important for people to know. But just before I yes, I'm a serving Lieutenant General, and I share my school with Naren and my college with Anjali. So we make a fantastic threesome to get things going for this afternoon with all of you, and I look forward to this. But I think it would be very unfair if we don't know a little more about this author, Dr. Narendra Chirmule, who's gone to book writing. I have the pleasure of having known him from school days, where he always had his finger in multiple pies while studying as a science, this thing in at RDSO. He was playing the tabla, playing table tennis, and Naren, if I remember right, whistling fantastically all the tunes. Not just for the girls. Yeah.
I'm glad you said that and I didn't have to.
Yeah. So from that, you moved a long way, and it's been great to remain in touch and watch you go up for that path of a professional from something, you followed your dreams, your passions. And not just the book, but we'd love to know a little more about what went into writing, why the book, what was your thought process, and this journey which you have traveled. So I think today we'd like to know a little of all that when we chat.
Thank you, Anjali, and thank you, Madhuri. It's such a pleasure. It's so fun to be able to do this special day with both of you. So what I've done is, because I know no better, I need to show slides. So Chetna, you might have to give me permission to share the slides. So I see a lot of my friends and family on the call. So I'm so happy that everybody has taken the time to join. So as soon as Chetna gives me the permission, I will be able to show you the slides.
If I may have a word, Naren.
Please do.
She's waiting, since we are waiting for Chetna. I just wondered whether you noticed, you said in the book that you have the number 7, which you love. It's also your date of birth, right? So did you notice that in the book, Good Genes Gone Bad, GGGB, G is the 7th letter of the alphabet?
I didn't notice that. Wow, this is absolutely absurd and
That's
crazy.
another 7 to your forwards of what
Yes.
all, philosophy of the number 7. Fantastic,
Yeah.
Anjali. Great one.
Yeah. No, wonderful, wonderful. So I'm going to share my slide just so that I put it in perspective. I'll take about 5 minutes to show these slides. So the book I have written is called Good Genes Gone Bad, and we'll talk about or the title also. The central thought of the book is a history of vaccines and biologics development through my own personal experiences. It's almost a little bit of like an autobiography of my experiences. And why this book? This, as I said, the book is my experience in drug development in various places, and it is all about failures that I experienced, and also the drugs which failed, colossal failures. In all the cases, failures were followed by phenomenal successes. And indeed, were it not for these failures, the path to success would have followed a very hazy, long-winded, and tiresome process. And to appreciate the success of drugs that are so successful today, I thought it was important to chronicle and recognize the contributions that come from. So that's sort of the reason I wrote the book. I've been, for lack of a better word, infatuated with this term called inflection points. And Madhuri, you might, and Anjali, of course, remembers when I came to AFMC, that's all I talked about, and how to find your inflection points in your career. So one of the things that I've done in the book, which I find very useful, is because this is the way sort of I live my life also. The work-life balance between all the work that I do and the play things that I do, so-called play, the art and music that I do, is not separate. It's interweaved within itself. So a lot of things that I have written in the book are soft skills, things that I have learned along the way of how to improve yourself. I also call it self-awareness. So, for example, I've written about what are the inflection points that one needs to make a difference in their career. Things like that are listed in this slide. So not just the scientific part, but also soft skills are weaved through the book. And that is the book. Kiran was very kind to write a foreword for the book. I've known her for several years now, and it's a really honest and true reflection of her own failures that she has written in the book. And Siddharth is a good friend, and he read the book earlier, and so did Madhuri and others who have read the book and given me very good comments. So I write the story of drug development as you can imagine, 7 chapters, and then I draw these figures. And Gauri Divte, who's my niece, has made all the diagrams in it. And the way I've drawn these figures is by looking at the timeline of all the successes and failures of these drugs, and then seeing what happens over time. And as you can see in the story of gene therapy, there's a death in a clinical trial, mutations that result in clinical trials, which are really bad events in the process of drug, but ultimately everything rises up. These are the 7 chapters: haemophilia, rotavirus, AIDS, the story of immunotherapy, cell therapy, gene therapy, and then my own experiences, biotechnology. And then last year, since it was COVID year, I wrote a separate chapter on COVID, and you notice that I've not numbered that chapter. And one thing I want to mention is my dear friend Murli, who has taught me so much about life itself, and who's living with hemophilia, is on the call, and he is the protagonist of story number 1. This is Murli. He has hemophilia. And I've written my experiences, and he and I wrote the book together. I must say that first chapter, he is the person with the most positive attitude. So then I've written the story of gene therapy, all its positives and negatives, until the approval of the recent Luxturna, which is the gene therapy drug. I also then there is a failure of the drug. I talk about, like, this is Jim Wilson, is a very dear friend and also a mentor. I write stories about these scientists who experienced failures and how they personally overcame these failures and what their learnings were in the book also, and by a lot of conversations with them and their experiences. So, and then this is the ultimate story of gene therapy where we got success. Jean Bennett had played a big role in the development of gene therapy. I tell her story. And then finally, for bringing it back to hemophilia, Kathy High, for example, has done a lot of work in getting hemophilia gene therapy to where it is today. And this is Murli and me meeting in August in Chicago, just as a completion of that story. And then I'm so happy the book is published, and I'm looking forward to talking about it today and all of you hearing. Yeah, so let's open it up for any discussion. Anjali, maybe you can start and...
Yes. I mentioned about the seventh letter. Then there's another thing that I saw, that the design of the cover of the book, I have the book here.
Yeah.
The cover of the book looks a lot like the 4 nucleotides which make up the gene, A, C, T, and G. You know, it's as if they are scattered in the making of the gene.
Yeah.
Story of the nucleotides. Tell us the story of immunotherapy that you wrote in the book.
Yeah. I mean, good question. So one of the things that has helped me write the book a lot is, especially in the last 3 years after I retired out of Biocon, and maybe even a little bit before, I have been teaching quite a bit. I've been teaching online in various universities around the world. And because of that, I'm learning so much more. While teaching, I'm learning so much more. So, and I'm also learning, I'm articulating the ability to tell stories. Excuse me, there's some background noise, Chetna. There's someone on, you might have to unmute. Anyway, unless it's mine. All right. The story of immunotherapy is transformational, because immunotherapy is the aspect of gene therapy, of treatment of cancer, which is really curing cancer. We can actually now talk about curing cancer in certain situations. So that's why I wrote the story of immunotherapy. It is by a defining moment of studying a specific molecule on the T cell, which is called CTLA4, which, when you block its activity, you can enhance T cell function. And harnessing the power of that receptor won Jim Allison the Nobel Prize a couple of years ago. And that what has transformed the field of immunotherapy and resulted in drugs which are able to cure cancer. That's what the story is about. Just 1 second.
Can I come in?
Are you getting background?
I think we need to unmute, mute all except, the ones who are holding. I think now we're good to go. Have we been able to do that? Chetna, have you been able to mute everyone?
Yes, everybody else is on mute.
Yeah.
Yeah.
Some sound coming in from somewhere, as if there's a TV or a radio, giving this thing. Anyway, coming back to the... Naren, I particularly enjoyed reading your reflection right at the beginning of the book. You've beautifully brought out your journey and reflected on the progress along the way. And in that, there was one point in our journey together while we were in our respective fields. I remember you coming to AFMC and talking about this adenovirus vector and how you'd work. Can you tell us a little bit about that? Because I thought that Very fascinating,
through
especially
inflammation.
now.
Cytokines increase gut permeability, so there's more leak there. So this local immune response
Who
is
is this
inhibited. Increases
Natesh?
lipopolysaccharides.
Natesh is the
Reduces
person who
insulin
needs to be
sensitivity.
muted.
It also apparently makes people more hungry, so actually makes them eat more. And this directly
Nagesh and
contributes to
Natesh needs to be
So
muted.
in addition to this, people with a healthy gut have a more
Yeah, now I think we're good to go. So I want to know a little bit, and please can you share that, because that was something we had interacted even then. And today it's become so important with the vaccines for COVID.
Yeah. So thank you. I got distracted with that noise. There was a question about a little bit about adenoviruses.
Yes. Adenovirus and the vector, the way you described about it. Yes.
Yeah. So the story about gene therapy starts in 1980s or so, when it was realized that to be able to transduce genes into a cell, you need the properties of a virus. The viruses evolutionary are very efficient in taking genes from outside the cell to inside the cell. When you have just naked DNA or RNA, it's very difficult to put that into the cell. So the property of the virus is to take the gene, infect the cell, and transport that DNA or RNA of the virus into the nucleus of the cell. That property of the virus was harnessed by people who were trying to do gene therapy in the 1980s, 80s and 90s. And that's when adenoviral vectors became very popular. Adenovirus is a virus that causes the common cold in all of us. And so what's done is the virus is taken and its genome is engineered, which is basically the parts of the gene are cut, and some portion of the virus is removed and replaced with a portion of any protein that you want to express inside a cell. So in that case, it was a gene for a gene defect in a genetic disease called ornithine transcarbamylase. But now, for example, it is for the Chadox, for example, the Covishield vaccine. They take the adenovirus and put the spike protein gene into the adenovirus. So it makes the vaccine. That time it was the adenovirus, and so that's how adenoviral vectors are used.
But then before we got this CRISPR technique, how was the splicing and actually this thing done?
Yeah, so good question. Molecular biology is a little bit like cooking. You add A, you add B, and then C shows up. You add your haldi and you add your chalk, and suddenly your dal tastes really good. And literally, it is like molecular biology is like that, because it is a little bit of a leap of faith, because you can't see anything. You have this tube in which you think you have DNA, and in this tube in which you think you have the enzyme. You add both of them, and something cuts something, and then something happens. So it's a little bit of magic, but the science behind it is very deep.
Yeah. So do you hope to bring that thrill across to the readers of this book? I hope
yes.
what they can look forward to when they read the book.
I tried to weave that story. And Anjali, you may enjoy one part of the story which I wrote in your household, where you are the cook. Maybe, I don't know if you want to talk about that a little bit.
That's right. That's right. In fact, I was going to say that was one of the things which I particularly liked about the book, the analogies that you give as to how the methods are, step-by-step methodology, and each process follows the next one. So you get the analogy of my keeping things out for the cook to make, then someone else at another time keeping out the ingredients, and the third person reading it. And with each of them, you had labeled it with the particular organelle in the cell which took that forward, so as to have the complete product. And each one does not know about the sort of existence. There's no actual connection between the two of them. So that analogy was very good. Perhaps you could just run through that little bit. It would give an idea.
So, Anjali, if it's okay, rather than tell the actual thing that I've written in the book, I'll tell you what I learned in the process of writing the book. So, so what happened was I started writing the book in about, seriously, I started writing the book, meaning I had written a lot earlier, but when I said, okay, now I'm going to start writing the book, it was sort of February 2019. And so it took me 2 years or so to write the full book. And when I wrote the book and submitted to Gurveen, who's my editor, Gurveen Chadda, she said it's quite difficult to read. I mean, there's still a lot of technicalities and needs to be simplified. But so the next year, I spent trying to simplify the book. In the COVID years, I simplified the book. So one lesson that I learned, and that might be useful for anyone who's trying to write a book on a technical subject, which you want to make simple. 3 things that I found very useful. One is when I'm writing the story, what was I doing during that story? Like, for example, when I write the story of haemophilia, and I'm writing, that haemophilia is caused by this gene mutation and all that, it was really, really helpful to sit down with Murli and write the story of haemophilia as a personal story of somebody's journey. So the personal touch helped write the story, right? And Murli, thank you so much for being part of this, right? Then the second, that was what am I doing? The second thing that Gurveed suggested was what is happening around the world at that time. So, bring people to that era by telling anecdotes and stories of what's happening at that time. And the third thing was the analogies. So if you can find an analogy to explain certain things, then it's always better. So those, instead of telling what the analogy is, Anjali, these are the 3 lessons I learned to how to simplify scientific writing.
Right. One more analogy which you had used was also very interesting, particularly since Madhuri is from the army and she is here. You had given, the war when a terrorist enters a cell and you say that that's the antigen, and
Yes.
Finally it is up to the general to take a call as to which forces to call out. And we've got a general amongst us. So I thought particularly Madhuri will thoroughly enjoy that bit of writing. That it's a very interesting way, and it sticks. The learning from that sticks because you bring in the various, Air Force and the Army into that, and people can understand what it means. Immunology is not a simple subject, but to demystify it for the public has been very important, and you've done it very successfully in this book. Though I must add that the HIV story I found was the toughest. And you started your career with HIV and AIDS, did you not, Naren?
Yes.
What I want to ask you is, how do scientists, find the motivation to keep going despite so many difficulties? And in the in the research of in the research and then taking it forward to drug development, because Madhuri is also into research now. So I thought this would be interesting because the 3 of us could discuss that. How do they get it going? When does it start? Does it require a mindset?
Madhuri, maybe you can start and then I'll follow.
I just wanted to kind of jump in and say, I think, the 3 of us talking on this is exactly the triad that is required today. We have Naren here, who's been a fantastic scientist, gone the path of research, whether it was his early days, he's brought out even the work he's done on the leprosy vaccine back in India. And it was really way ahead of its time, where no one really thought of that vaccine, and then went on to do a PhD and then a postdoc and continued with his journey with these biologicals. And it's a very valid point. I think Murali's, this thing has got unmuted. So in this journey along the way, Naren, what Anjali's point is well taken. There are clinicians, the motivation for them is to see treatment translate into a patient doing well. There's immediate gratification and you're seeing a translational value. But for a scientist, you don't have the results, and not everything goes up into, successes. there are huge failures. And what I particularly liked about your book is reflecting and talking of these failures in whether you call it the inflection point or the failures which led to big discoveries, which we know. And therefore, when there's a failure and you do not have an incentive ahead of you in the form of a patient whom you're caring for, I think it requires tremendous amount of motivation and what you've explained in your this thing of attitude and passion. Absolutely, I think that's a very important point. So I think this triad today we are here. Anjali is the clinician, you are the researcher, I'm the clinician researcher. So I think we need this. There are certain missing links when it comes to education, research, and health, and making these big discoveries. So the inflection points do not happen because we don't have closer collaboration. Do you think that is the case, Naren?
Yeah, I think that you hit the nail on the head, and I think now in retrospect that I've written the book and I've written the 70 chapters that I've written about, I really think that it is the attitude and the ability to tolerate failures and to deal with failures and really bad failures is one of the characteristics of the ability to do research. Because you do 10 experiments, one of them works. And you have to have the persistence to be able to, almost like a mad dog, just go after it and try to solve the problems. Because many times the rewards of that are very, very small, very small. You just make incremental gains in huge efforts that you put. But it is those little incremental gains that ultimately add up to big stories, right? And we, in the news media, we only hear the big story. So Bruce Levine, who is sort of the pioneer of what is called CAR T-cell therapy, which is transforming, is also a good friend of mine, and he read the book. And one of the statements he actually says in his lectures is the overnight success of CAR T-cell therapy took 30 years. I love that sentence.
Yes, absolutely. So that's why my specific, compliments to you for bringing out this, because you have put it, it's not just important of having brought out these stories, but simplified it for everyone to understand. Because gene therapy sounds very utopian, very fancy, extraterrestrial, things which you feel is beyond dreams of this. So bringing it down into simple language, making it into a story-like fashion, and what you described of Gurleen giving you those inputs and you using them effectively. Because the moment it translates to a life story, the moment I can relate to it, to events which I am aware of, I think the way those have been woven into these stories makes them very good reading. I totally agree, and compliments.
Absolutely, Madhuri. In fact, the way you said weaving it into a story, I think another thing that Naren I have observed has been reading, regularly and in detail the Nature magazine. The discoveries which are made all over the world. In the failures, you find some discoveries made somewhere which fit into your line of thinking, and you find maybe that could be a solution, to stitch that together with your story and to take it forward ultimately to a successful outcome. That is really hard, and to find those inflection points of this is the place where this particular discovery made a difference. And just as you said, the CAR T cell, similarly, there are so many in Naren's book, which the greatness is in finding that particular turning point and to be able to go forward with that. That is not easy. Actually, it's a philosophical question. How do you find the inflection point of a place and identify it as what, in fact, you've used that in almost all your stories. You've identified an inflection point.
Yeah. No, I think that's a great question, and there is no simple answer to it. I think it is the process of finding some inflection point. That there's not usually one. is again the process of research, right? It is the methodology of how you look for things. You find many things, you just keep writing what are the possibilities. Like for example, if you think about, if I said CAR T cell was the inflection point for immunotherapy, right? Then okay, it could be CAR T cells, it could also be it could be this CTLA4 antigen, it could be adenoviral vector, it could be this, that, on the, you write everything down. You write everything down, that all the possibilities of what that inflection point could be. And for each one of them, you explore further. Say, okay, if it was the adenovirus, is it this, does it meet the criteria of what I think an inflection point should be? I've got 100 points that could be inflection points. I evaluate each of them, whether they should be inflection points. And then with pros and cons, saying, okay, this is the most likely inflection point. So it's a detailed methodology. It's not like just you look and for me, it's not like you look and find it.
So Naren, can you say that you know it was the inflection point, probably more in hindsight, that for this was the inflection point? You've done your reflection, you've done your analysis. That's what the scientific mind is trained to do. So you look at all the probabilities and then choose the path, and then the path which goes the correct way, you say, okay, that was the correct decision.
You hit the nail on the head. It has to be retrospective.
Yeah.
And then you learn.
I think you make an understatement by saying you hit on 10, you knock on 10 doors and one opens. I think in research, it's hundreds of doors, whether it's for getting the grants, whether it's for the funding, whether it's your outcomes, whether it's your publications. You keep knocking on hundreds of doors every time. And that's where persistence is.
It reminds me of the drug development story that Naren has done with hundreds of people, including in my organization, I think also in AFMC, CGHS and AFMC, including physicians. People don't know what goes behind the drug development that occurs. You cannot suddenly say, in fact, the way Naren has done it, you choose a disease, you say you want to cure, and what do you think, where will you start? Where do you get, what is the molecule which is going to cure that disease? So where does that come from? That comes from pure science study in academia. So Naren says you have to go and, if you are a commercial venture, you have to go and buy that from that academic institution, because they have the rights of discovery. So I think, is that the reason why when it's so difficult to find out which is the specific drug, you will have to test hundreds and thousands to be able to treat one disease, to take it further. It's not at all easy to find those molecules. Is that the reason why we are doing biosimilars, which is in India? Because everyone need not reinvent the wheel, so to say. So is that the reason why we are doing biosimilars in India in a big way now?
Well, yeah, in a way, yes. I think the Indian biopharmaceutical industry is, maybe I'll speak a little bit about the industry itself in India, and then maybe this could be a good segue to discussion, Madhuri, with what education in this area needs to be. And then after that, maybe we can open it up for questions from the audience, if that's okay, Anjali. So the Indian pharmaceutical industry, is in, so maybe if we have to put a timeline around it, really the 2008, which is when the WHO treaty was signed by India, where we would honor the patents of the world. That was a sort of line in the sand, before which, before 2008, there were a lot of companies who developed drugs like Lipitor and other statin drugs, which was still in patent in the rest of the world, but could be developed in India because of the rules and regulations in India. And so the Indian pharmaceutical companies got the heads up in developing these generic molecules and became world leaders. In fact, today, greater than 40% of drugs sold in the world are made in India in some way, form or the other. right? Either the raw materials or the API, or so those things are made in India. It's a huge, huge, huge success story for generics in India. All the companies like Cipla, Dr. Reddy's, or Bindo, you can name them. There are many, many, many companies. So India, the Indian diaspora of companies really learned how to make generic drugs really well. They would take a molecule that was approved in the US, which showed, or in the rest of the world, that showed to be working on a particular disease very well. They could reverse engineer that drug and develop the drug as a generic molecule, and it was getting approved, and it could be sold at much lower prices and became much cheaper for people to buy it.
Yeah.
But still, it's not an easy process, I think.
It's definitely not an easy process. It's definitely tough, but they've mastered the art of doing it. Similarly, biologics, which are drugs which are made in biological systems, for example, cells will express the protein which makes the drug. The generic version of the biologic is called the biosimilar. So that it has a fancy name, but it is essentially a generic of a biologic. Again, it's very difficult to make. Some people say it's even more difficult to make than the original drug, because you have to make it exactly the same as the original drug. So that's what biosimilars are. And I think the future of biotechnology in India has to be to make novel drugs. I think that's where, that's a segue to Madhuri. I want to have a discussion with Madhuri, and we heard Madhuri's comment. What would it take to develop clinical scientists in India?
I think we'll get on with the education and the science, this thing, because I think we have reached the I think this is the inflection point for research education, especially in biotechnology. I think we are really at the threshold of, and we can go in for absolutely disruptive, rapid progress if we get our act together. I mean, just before I take on this education and there's one thing that I'm particularly fascinated and I want you to tell all the listeners about, is being a pediatrician, and because India had a big role with the rotavirus vaccine. So I thought, how while you were working on the rotavirus there, India had its own, and it was after we set up our Department of Biotechnology. I mean, this was one of those first vaccines where we started from scratch. The actual the virus for the Indian, this thing was picked up from a strain from the All India Institute of Medical Sciences and went on to develop the vaccine. So this was why, for two reasons, it's fascinating. One, as a pediatrician and diarrhea being such an important thing. Second, because what you pointed out, about 40% of the drugs, today, every sixth baby in the world is given a vaccine made in India. This is the capability we have reached. And that is why I say we are just at the threshold of great advances, because the time is right. So I'd like to take that further discussion after we hear from you on this story of the rotavirus a little bit more.
Yeah. So one of the I guess events in my life was after finishing my education in India in 1987. I went to the United States, and I lived there for 30 years. So for better or for worse, my worldview of everything that was happening was very US centric. I was not seeing what's happening in the rest of the world. I mean, my eyes were almost closed. So all the work that went on, amazing work that went on in India, in Krishna Ella's group in Bharat Biotech, where they developed the rotavirus vaccine, was not mainstream visible to me until much later. So I learned about that vaccine much later, and I've written a little bit about that in the book. But developing a rotavirus vaccine is extremely difficult, and that part of the story is written in the book. And one of the side effects of the vaccine, the earlier vaccine that failed, was a situation that occurred in children called intussusception, where the intestine telescopes into itself and babies die. A normal baby, like otherwise completely normal baby, give the vaccine and the baby dies. That risk is unacceptable. And you know, the social aspect of the vaccine, which I have not written a lot about but touched upon it, is the fact that the risk of the risk of any adverse event that occurs and kills people, for example, with a drug is much less tolerated in let's say in US, because I know more about US, and maybe the risk tolerance of a few patients dying in India, in a country like India, may be higher, given that the benefit is so much larger. So the risk benefit ratio in different places may be different. And that's a social discussion on vaccines that, you know, that itself is a book. That's a book in itself.
Yeah.
But yeah, so
But
so
do you
that's,
think, yeah, so this, when you were talking of the biologicals, I think, again, India, because see, we've used for the first time, we've produced the DNA vaccine, the Cadila one, which is going to be used for
yes,
children,
first.
COVID, because it's been a known technology. But I mean, it's been translated to, and we are ready to sort of have bring out this vaccine. And another aspect I'd like to know, because you've covered COVID itself, I think it's been fascinating from it's probably the first time in history that, you know, the virus from, we'd never heard of a Corona-19 virus. And within a year, we came out with a vaccine for it. And how do you think in your journey or in your inflection point, I mean, this mRNA technique, because that's the first time in the world such a thing has been used.
Yeah. So again, I haven't written much about the history of mRNA in the book. It is, you know, the Drew Weissman.
I'm giving you food for the next book.
I love it.
I can't stop at 7.
I love it. I love it. And you know, I think
good genes, you can call them good genes getting better.
Getting better. That's so true. You already have the book title. It just takes 3 years to write the book and a lot of sweat. So yeah, no, these are excellent questions. You know, there are a few questions in chat, Madhuri and Anjali.
Yes. And I thought I must this thing out, because there's a beautiful question from Hemant, which says that it says, which story out of the 7 is the first point of inspiration for you to think about writing a book?
You know, no question, the story about Murli. I mean, Murli and I have been talking about this book for 10 years. You know, he is the inspiration. And Murli, I don't know if you can put your video on. If you can, that'll be great. I would really want to introduce you. Are you on, Murli?
Yeah.
Are you able to be put on?
We want to see you, Murli, because he's made a wonderful comment here, which says, Naren is not a jack, but a true master of his trade. Look forward to reading the book. Absolutely. I think it's something everyone, not just on this forum, but every student of biotechnology should be looking forward to reading this.
Thank you, Madhuri. So let me introduce Murli to all of you. Murli is a dear friend of mine, and he and I did his PhD together. I won't elaborate on the story that I told about him, but you can read about him in the book. But Murli, if you just want to add a few cents of, you know, you read the first chapter, so maybe you can comment a little bit.
Hello, everybody. Yeah, I've been listening for the whole session, and especially hello to Anjali, who I have met, and hello, Madhuri also, and the rest of you. Yeah, I'm here in the Chicago area, and Naren and I actually go back a long way. And as far as the book itself, we've been working on this. Actually, I know Naren has been working on this for at least 2 years. And then he obviously wanted to start with haemophilia because he knows me quite well. So he would send me drafts and questions. It started as a question answer, and he put it together as a draft, and then we met in 2019, and we actually had almost like an interview type of question over lunch, breakfast, spent a lot of time together. And that is when I realized, like, I have to actually reflect on, my own history to be, actually reflected in the book itself. So I had to think back quite into the detail, and he was very serious and detailed in his questions. It got more and more details, and he was able to therefore include all those details in the question, and somehow or the other, that's Naren's skill. It's all Naren there. Put it in such a way that the flow of the events and all the missing parts, everything came out. I haven't actually seen the very last draft before he sent it in, and then he told me, like, you know what, I don't want you to see everything because I would like you to to read the book. And I said, yes, there's no point, there's no value to me reading the whole book from you until in chapters, until I get hold of the book. So I'm really looking forward. I'm so proud of you, Naren, and so happy that it's finally done, and we are having this conversation about the book itself. So we'll look forward to the second book as we the part 2 sequel to this book, and wish you the best, Naren.
Thank you. Thank you.
I'm just coming here.
Naren said there was no mentor, but he did have one for this book. Great, Naren, that's way going. But Tanya says, how do you overcome blank page anxiety when you sit down to write? Did you have it?
Yeah. Lot of anxiety of writing on blank pages. But Tanaya, I think one of the ways to overcome that is to write anything. Just write anything, even if it's gibberish. Just write. I think Ajit is here. Ajit Datar is here on the call, and he told me one story one day. And Ajit, maybe we'll confirm whether it's true or not. But I tell this story so many times that it may be actually true. So there's a small village in Maharashtra, in where 1 year from that school, a lot of kids came into the distinction. They got, like, a huge number of students became, got very high rank in the 10th class exam, SSC exam. And so when they looked at what happened, and suddenly invited one small tiny village school in a tiny village in Maharashtra get this, it turns out that teacher in that school made the students the whole year sit down and write something for 4 hours every day. They would go to school, and for 4 hours they would write anything. I'll tell you in Hindi, Sholi ki story likho, lekin likho 4 ghanta. If 4 hours you have to sit down and write anything, right? Because what happens is you study for the exam, and you've never practiced sitting down for 4 hours and writing. That's what the exam is about. Right?
Good one.
So, Tanaya, so the only way to overcome is to write. Just write any damn thing. So later on, some good things may come.
Yeah. One good thing of Murli, which pointed out, Naren, was that you went into all the details. And I think that's one thing, collecting data, being specific, and actually making only true collection of data without falsification, that honesty and integrity which is required comes across in the stories, because you can't paste over anything. You need to have authentic data, and that is where I think inculcation in school about this sort of thing, writing and being true to yourself and to the facts that you're writing, I think that's very important.
Anjali, I would second you on that, on the research that Naren has gone into. So it's not just a fairy tale story that he's woven, but there's a lot of research that has gone in, a lot of hard facts. It takes the reader through narrations which you can relate to, and you come out very knowledgeable. It's not just that you enjoyed reading, but you come out knowledgeable. So I think that's very important.
In this particular time, during this corona times, COVID-19, the general public has developed an interest in immunology, and they probably don't even know that it's called immunology. Everyone has an opinion on the symptoms of the disease and how it should be managed and which vaccine is good, and should I take this vaccine or the other one, And then if there are side effects, they say, they have unrealistic expectations of a perfect product, which is normally takes about 15 to 20 years to develop, and people would like it to be perfect right away. I think the book you have brought out very well. What is the complexity in making a vaccine? It's a very, very difficult process. And the allegations that people make, especially now, people are saying Covishield in Britain is not permitting Covishield takers to come into Britain and not have quarantine. There are so many things in those details. These are totally unnecessary. This vaccine hesitancy that people have, as if they can decide. It is science that decides, and they have documentation. They take all the possible precautions to do that. You brought that out in your vaccine stories, as well as in the last chapter for COVID-19. I don't know if there are any questions with regard to.
There is. Hemant wants to know that if there's any story that you have not captured in this book, if yes, and given an edit option in time, what story would you like to add? So now, I thought I was the only one, but there are others. Naren, your journey has just begun as a writer.
True. But to answer Hemant's question, Hemant, you probably know about this. Is there's a story that is brewing right now in the literature on the treatment for Alzheimer's. So I would have loved to write an Alzheimer's story because it's such an important disease these days. But I don't know much about the disease myself. It'll take a lot of reading and studying about Alzheimer's, but that's one story I would have loved to tell.
That's very important. And Purushottam has a question that I've heard that melting of glaciers may liberate viruses that are novel to human species. How do you feel we can handle these?
Oh my God! First of all, I didn't know about that, and I don't know how to handle these. I think I think we'll have to go through systematically through trying to find out. No, thank you for the information. I'll try to look it up.
There's Ashutosh who's asking a question here that even if the results are not 100%, how is it decided if it has reached the desired effects? I think that's a very good question on, it's the whole thing about designing a study and what is your endpoint. I think that is a very important aspect brought out.
Yeah.
And Rajiv wants to know, which is your eureka moment of all these journeys?
That's a good question. I think the eureka moment was literally the studies. It wasn't a moment. It was a span of time. It was probably in the 1990s when the story of the immunotherapy unfolded. There was a molecule called CTLA4, which I talk about.
Yes.
You can read more about it in the book.
Yeah.
For the longest time, it was thought that the CTLA4 molecule was stimulating T cells to induce their thing. When Jim Allison's research suggested that it is not a stimulator, but it's actually an inhibitor, that was oh my God, that's fantastic. But Rajiv, there are in research, there are so many. That's what's the fun of doing research. You do an experiment because you don't know what the result outcome is. Every experiment is eureka result.
Yeah.
Because if you knew these.
I would answer it for you, Naren, between Rajiv and me. I think for you personally, eureka moment for this is when you suddenly got that thing, I'm going to write a book. I mean, imagine changing directions. So, from scientists, research, scientific publications, this is something so different. And this is such a beautiful balance of your right brain and your left brain.
Yeah.
So I think that I
through
would
that.
give
Yes.
this as your eureka moment. So that's perfect.
I think that's a great one. That's a great one, Madhuri.
Yeah. So
And-
Suman has a question from Biocon. I'm yet to read the book. If Amazon could deliver the right book, I would have read first 50 pages by now. How do you manage to get so much done at the same time? Please talk a few sentences about your time management.
Well, I'm going to tell you one thing. There's a course that I took several years ago by a company called Calm Achievers, and I don't mean this by advertising. It is really a good course. It's called Getting Things Done. There's a book called written by David Allen, and they train you on how to get things done. It is a transformative course. I did that course, and literally after doing that course, I'm able to do a lot of different things. So in addition, you have to have the aptitude to want to do many things, but that book is quite transformative.
I mean, it's little away from the book, but you have also started this in your reflective, this thing on your initial letter. You said, I mean, I didn't really have a mentor. And I thought that it has really got you thinking, because you're taking on a mentorship role very seriously and doing a lot on that. So would you like to share something on your role or initiative as being a mentor?
That's a good point, Madhuri. And you're absolutely right. In an earlier comment, you had said it's not like we don't have mentors, and we probably have mentors in our lives. We don't know they are mentors because we've not thought about them that way. We just think that they are helping us, and therefore, they are helping us. We haven't defined that they are mentors. Like, even in our school, Ravinder Soni is asking some questions. We were sort of mentors to each other. Remember the class we used to take over the school, one of those on the Teachers' Day, and Ravinder Soni used to be principal, and he used to put his legs on the table and all that. We were all mentors to each other, but we didn't know that. But because I've sort of reflected on the fact that mentor is one, to have a good mentor or a couple of mentors for yourself is transformative for your career or your life. And so it is your responsibility to go and seek out a mentor. A mentor is not going to find you. You have to be the mentor. You have to go and seek out your mentor. And so what I have been doing in the last COVID year, in this last year, is I've been having these Zoom sessions every week with various groups of people who come in, and we chat about a lot of different things. And Madhuri, you've come and talked to some of the students in that group. It enables people to just talk about their problems, share problems with each other, and then ultimately come to their own solutions. Everyone has to find their own solutions. What I've also learned from reading, writing people's characters, like I told you about Jim Wilson, Jean Bennett, all of these people who I've written about, you can't really follow what exactly they did. You have to see what they did, incorporate what good and bad that you might have learned from that, and make your own journey. Your own journey is very different. So the mentor can't tell you what to, mentor can't get you the next job. The mentor can't, help you do the best thing. Mentor can only be there. It's like a mirror. So long-winded answer for that mentor, but I'm really passionate about the concept of mentoring.
Yeah. So Ravinder wants to know, when do we expect the next book? But before we ask the next book, I think you must share with everyone, is it immediately, the moment we finish with this dialogue, can we start ordering on Amazon?
I believe the book is available on Amazon. I don't know if Sumangala is online and if she can comment a little bit on that.
I will tell you, I pre-ordered the book. It did not come. Then I ordered on Amazon and it came the next day. This is it.
Lovely.
Yeah, and I'm going to read a specific line, especially for Ravindra Soni. Okay, it's in the epilogue.
On page?
The last page, yes. Will there be a follow-up book? Yes, but maybe after I get some feedback on this one. That's for you, Ravindra. Now you read it and give the feedback.
Yes, good one, good one.
Thank you for answering that question, Anjali.
So Naren, we've been reading, so, and there are lots of people more who are going to read, I'm sure, in this meeting, and I hope you have more meetings, because it really, it's not just this thing, but you get a little oversight and a curiosity. So this, I'm sure this appetizer has been a good for the main course.
Wonderfully well said. Thank you so much, Anjali, and thank you so much, Madhuri, for spending this time and talking about the book.
Yeah.
It's been an absolute pleasure. There are plenty of things we can talk about, but it can be done over other sessions. There's lots in the book to explore, and each one is a big chapter by itself, worth reading and digesting. Thank you, Naren, for writing the book, and we are so proud of you.
Thank you. Thanks, and thanks for having me part of this wonderful story. So, and all the best for all your future endeavors. And yes, I'm ordering a hard copy, because this thing which you sent for us to read before is not good enough. I'm still old school. I like a hardbound copy in my hand, which I can sit back and enjoy reading. And I look forward to doing just that. And I'm sure all the people would do the same here. Thank you so much.
Thank you very much. Thank you, everyone on the call, and thank you for listening in. Bye.